Hi everyone, in this PowerPoint presentation, I will be giving you a general overview of animal cell structures and with a focus on the different types of organelles and their main functions. Let's get started. Now, cells are the smallest living units of an organism. All cells, they have three things in common, no matter what type of cell they are. So all cells have a membrane, which separates the inside of the cell from its environment. They will have a cytoplasm, which is this jelly-like fluid inside of the cell. And they will have a DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is what we call the eukaryotic cells, and they have organelles, which includes the nucleus and other special parts. Eukaryotic cells, they are more advanced, complex cells, such as those that are found in plants and animals. The second category is what we call the prokaryotic cells, and these, they don't have a nucleus or membrane-enclosed organelles. They do have the genetic material, but it's not contained within a nucleus. As we can see over here, the prokaryotic cells, they're always one-celled or unicellular organisms, such as bacteria, which is shown on this image. The cell consists of a cytoplasm, which is made up of cytosol plus organelles that are enclosed or surrounded by a plasma membrane. The plasma membrane is made up of a lipid bilayer, which we will cover in more details in the next learning outcome. But just to cover a little bit of the function of the plasma membrane, it will isolate, protect, support, and control what goes in and out of the cell. And depending on the type of cell, it even has a role of detecting sensation if, for example, it is a neuronal cell or a cell of the nervous system. Now, in contrast, the cytoplasm is going to be made up of organelles and cytosol. Organelles are all of the colored structures that we are able to visualize on this image, where each will have a specific function, and we will cover each of them in more details on the next slides. How about the cytosol? What is the cytosol? It is going to be this jelly-like fluid component of the cytoplasm that contains dissolved nutrients, soluble and insoluble proteins, wastes, and ions. In fact, because cytosol is inside of the cell, it will contain high concentration of potassium ions, while the extracellular fluid or the fluid outside of the cell will contain a high concentration of sodium ions. And this is going to be important to keep in mind when talking about membrane potentials, which will come up through the semester. And therefore, the function of the cytosol is basically to distribute materials by diffusion. And this will allow the movement of molecules in a fluid from areas of high concentration to areas of low concentration. And we will see more about this later on in this lab. In addition, it is able to store glycogen, pigments, and other materials depending on the cell type. So what are organelles? Organelles basically means little organs, and organelles are going to be these specialized parts of the cells that have unique jobs to perform. Among the more important cell organelles are the nuclei, which is represented on this image by this big pink circle, and it will store the genetic information. Inside the nucleus, we find another organelle, which is the nucleolus. And surrounding the nucleus, these yellow structures are representing the endoplasmic reticulum. On each side of the endoplasmic reticulum, we find a couple of mitochondria, which are going to have a major role in the production of chemical energy. Then we have the Golgi apparatus, which will give rise to vesicles, represented here by these small lysosomes. And lastly, we find a peroxisome, which are organelles that are even smaller than lysosomes. Let's see in a little more details the functions of each of these organelles. Let's start with the nucleus, which is the control center of the cell. The nucleus will contain DNA, or the genetic material, and DNA dictates what the cell is going to do and how it's going to do it. So chromatin is this tangled, spread out form of DNA that's going to be found inside the nuclear envelope, which is also known as the nuclear membrane, represented in this image by those dark lines inside of the nucleus. When a cell is ready to divide or replicate, the DNA will condense or groups together into structures that are known as chromosomes. And the nucleus also contains a nucleolus, which is an organelle where ribosomes are going to be made, which are these tiny little round structures that we can see right over here, for example. So what are the functions of those ribosomes that were produced in the nucleolus of the nucleus? Well, after they leave the nucleus through the nuclear pores, which are these openings on the nuclear envelope, they will have the important job of synthesizing or making proteins. As we already know, outside the nucleus, the ribosomes and the rest of the organelles, they float around the cytosol. Therefore, these ribosomes, they may wander freely within the cytoplasm or attached to the endoplasmic reticulum, which are these large yellow structures that sometimes are simply referred to as ER. This means that there are two types of ER, what we call a rough ER, which have ribosomes attached to it, as we see over here. Notice all these tiny little dots on the ER, and the other type are known as the smooth ER, which will not have ribosomes attached to it. Makes sense, right? Rough, look rough with all those little organelles attached to it, and smooth looks smooth. As we can see from this image, it is going to be made up of this network of intracellular membranes that form these hollow tubes or flat sheets that form these rounded chambers that are actually known as the cistern. The ER have four main functions. First function would be the synthesis of lipids, proteins, and carbohydrates. And why would they want to manufacture all these products? Well, they have the perfect place to store them, in the cistern, which create these ideal pockets for storage of these manufactured products. Second would be storage. In addition to storing the products that is manufactured in the ER itself, the endoplasmic reticulum stores synthesized molecules and substances that are absorbed from the cytosol without affecting other cellular operations. Then it also helps with transport of substances, since they are able to travel from place to place within the cell inside the cistern of the ER. And lastly, it has a very important function of detoxification. Not that the other functions are not as important, but the ER absorbs drugs and toxins and neutralizes them with enzymes. And in this way, it is able to remove toxins from the body. Now, all these proteins and other materials that are stored and being transported inside the ER, they will emerge from the endoplasmic reticulum in small vesicles, and we can actually see a couple of them in this image, represented here by these two yellow circles, where the next organelle, which is the Golgi apparatus, will receive them. So that leads us into the next organelle, which is the Golgi apparatus, which can also be called the Golgi body or the Golgi complex. And it will consist of these flattened membrane discs that are called the cistern. A typical Golgi apparatus will consist of 5 to 6 of these structures, of these cisterns, that resemble a stack of dinner plates. In actively secreting cells, they have larger and more numerous cisterns than resting cells, which makes sense, right? Remember, everything should make sense. And the major functions of the Golgi apparatus are to package enzymes for the use in the cytosol, renew or modify the plasma membrane, and synthesize and package secretion. So let's look at all these functions in more details in the next slide. The first function, which is package of enzymes for use in the cytosol, among the vesicles that are packaged by the Golgi apparatus and remain in the cytosol are the lysosomes, which are filled with enzymes that break down engulfed foreign materials and pathogens as well as damaged membranous organelles. The second function, to renew or modify the plasma membrane, has to do with the fact that as the Golgi apparatus loses the membrane through the generation of these vesicles at the maturing phase, it actually gains membrane by the fusion of transport vesicles at the forming phase. So notice how we have a forming phase on this end and a maturing phase on this other end of the Golgi apparatus. So when Golgi-generated vesicles fuse with the plasma membrane, it adds to the surface area of the cell, balancing the membrane loss that occurs during endocytosis. And we will talk more about endocytosis later, but when endocytosis occurs, you're going to have molecules that are going to be going into the cell, so they're going to be getting part of the membrane into the cell, and therefore the cell loses part of the cell membrane. So there is this balance of the membrane that's being added onto and the membrane that's being removed to form the vesicles that are going into the cell. Over time, this process can change the sensitivity and the function of the cell. In an actively secreting cell, the change can be rapid, and the entire plasma membrane may be replaced every hour. Next and last, we have the synthesis and package of secretions, and this occurs at the maturing phase, therefore on the opposite side of the forming phase. And this occurs when vesicles are formed, and these vesicles will carry material away from the Golgi apparatus. Now, these vesicles containing material that will be secreted from the cell are called secretory vesicles. When a secretory vesicle fuses with the plasma membrane, its contents are going to be released into the extracellular fluid, as we can see right over here. And this process is known as exocytosis. So we have endocytosis occurring in the forming phase, and we have exocytosis occurring at the maturing phase of the Golgi apparatus. Mitochondria are these membrane-bound cell organelles. When we want to say the singular form, we say mitochondrion, that will generate most of the chemical energy that's needed to power the cell's biochemical reactions. It will produce ATP by breaking down organic molecules in a series of reactions that will consume oxygen and actually generate carbon dioxide in exchange. And this series of catabolic chemical reactions within the mitochondria produces about 95% of the energy that's needed to keep a cell alive. Also, notice how the mitochondria have an unusual double-layered membrane. There will be an outer membrane that surrounds the entire organelle and a second inner membrane that contains, as we can see, several folds. These folds are called cristae, and these cristae are important because they will increase the surface area for the organization of the enzymes that produce most of the ATP. And as you can imagine, the number of mitochondria in a cell will vary depending on the cell's energy demands. So for example, red blood cells, they lack mitochondria as they will obtain energy in other ways. They actually rely on glucose for their energy source. But skeletal muscle cells typically contain as many as 300 mitochondria. Makes sense, right? If you have to run, for example, you need a lot of energy. And this means your muscle cells will consume a lot of energy. Therefore, these cells need to have a lot of mitochondria. And the best characteristic of all is that it has the ability to self replicate. In other words, produce more of its own. Next, we arrive at the peroxisomes, and they are similar to lysosomes in the sense that they also carry enzymes. However, they are smaller than lysosomes and they carry different enzymes. Therefore, they will have different functions. Remember that the lysosomes were formed by budding off the Golgi apparatus. Now, the peroxisomes will contain enzymes that are formed by free ribosomes that are circulating in the cytoplasm and are then inserted into the membranes of the pre-existing peroxisomes. What does this mean? It means that new peroxisomes results from the cell recycling older pre-existing peroxisomes that no longer contain active enzymes. So the free ribosomes are circulating. They find a peroxisome that don't contain active enzymes, and they will insert new active enzymes into these old unused peroxisomes. Very interesting, right? And in general, enzymes are a substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions proceed without the enzyme itself being altered in the process. And because they're made by free ribosomes, they're usually composed of proteins. And they will work by actually decreasing the energy that is needed to complete a chemical reaction. But there will be certain external factors that might affect the efficiency of the enzyme's actions. For example, if there is a change in the temperature or in pH or even salinity, it might also depend on the concentration of the enzyme and or the concentration of the substrate. There might be some inhibitors, activators, or cofactors that might be present or even absent, and this might be interfering with the efficiency of each enzyme. Now, the enzymes within the peroxisomes, they perform a wide variety of cellular functions. One group of enzymes, oxidases, they break down organic compounds into hydrogen peroxide, which is toxic to the cells. And catalase, another enzyme within the peroxisomes, they then convert hydrogen peroxide to water and oxygen. So peroxisomes also absorb and break down fatty acids, and peroxisomes are most abundant in liver cells, which will remove and neutralize toxins that are absorbed in the digestive tract. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made mainly of a protein called actin. In terms of functions, the microfilaments will anchor the cytoskeleton to proteins of plasma membrane, more specific, the integral proteins, which we will talk more about when we cover the plasma membrane components. And this helps to stabilize the position of these membrane proteins. It also strengthens the cell and attaches the plasma membrane to the underlying cytoplasm. Also, the actin microfilaments interact with other microfilaments or larger structures that are composed of protein myosin, for example. This interaction allows part of the cell to move, or it actually allows changes to the shape of the entire cell. And microtubules, which are thin hollow tubes, like the name says, microtubules, so small tubes. Makes sense, right? And these tubes are built from the protein tubulin. With regards to the functions of the microtubules, there are several. As they form the main components of the cytoskeleton, they will be giving the cell strength and rigidity and also anchoring the major organelles. They also allow cells to change the shape and may help the cell to move. And because they attach to organelles, they help these move around the cell. Next, they will have an important role during cell division, which we will talk more when we cover this topic. And lastly, microtubules form structural components of organelles such as centrioles, cilia, and flagella, which we will see on the next couple of slides. A centriole is going to be this cylindrical structure that's composed of a short microtubule that are organized. Notice how they are not scattered throughout the cell. They actually form 9 groups of microtubules, and they will always be paired within a cell, meaning that there are always 2 centrioles, and they will be found in a specific region of the cytoplasm that is always close to the nucleus, known as the centrosome. Also notice how they are arranged at right angles of each other, and this disposition is going to be important to move the chromosomes into opposite directions within the cell during the cell division. Some cells have unique features. For example, the cells in the respiratory system, they're going to be lined with what we call cilia, as we can see right over here. These are going to be microscopic hair-like projections that can move in waves. This feature helps to trap inhaled particles in the air, and it also expels them when you cough. And we're going to talk more about this when we get to the respiratory system. Another unique feature is the flagella. Some bacteria have flagella, as we can see over here from this image. A flagellum is like a little tail that can help a cell move or propel itself. And the only human cell that has a flagellum is a sperm cell that's shown on this image over here. And it looks just like a tail.